This is a journey about a bubble boy and his journey to a normal life.
I think it's down here. Go ahead.
Welcome, Stephen.
Just turn it off.
Well, as you know, Stephen, you have SCIDs, or severe combined immune deficiency. This is the most severe of the primary immune deficiency diseases. The characteristics of your disease are the absence of T cells and, as a result, lack of B cell function as well.
But we think we can fix you with gene therapy, which is a technique for treatment of genetic disease in which a gene that is absent or defective is replaced by a healthy gene. And we can usually do this by replacing the abnormal gene for a normal gene through homologous recombination. So we're going to discuss a little more about what we want to do with you, and we'll come tell you later.
Well, for gene therapy to advance to its full potential, scientists had to discover the biological role of these individual genes and where the base pairs that make them up are located on the DNA. To address this, the Human Genome Project was started. It was a 15-year project, and its goal was to map the entire human genome. A genome map would clearly identify the location of all genes as well as the more than 3 billion base pairs that make them up. With the precise knowledge of gene locations and functions, we may be able to conquer or control diseases that have plagued humanity for centuries.
So, Ricky over there in the other room has had gene tests and biochemical tests to prove that he has SCID. So we need to identify the gene that's mutated and then find a healthy copy of that gene. Do you remember the kid that was born in 1968 and his mother thought he would have an immune deficiency because her child before had previously died, and so she put him in a bubble. And
yes, I do recall that.
They could never find a match for him, so he had to live in his bubble the whole life, and the press made him known as Bubble Boy.
That poor child.
I know. So we're not going to let that happen to Stephen.
Such a tragedy!
Well, Ricky, my colleague, Dr. Courtney, is going to explain to you how we're going to fix you with gene therapy. Dr. Courtney?
Alright, so first we are going to remove the cells from you, the patient. Then in the laboratory, a virus is going to be altered so that it cannot reproduce. A gene will then be inserted into the virus. The altered virus is mixed with cells from you, the patient. The cells from the patient become genetically altered.
Awesome.
The altered cells are injected into you, Ricky. The genetically altered cells produce the desired protein or hormone.
I have hormones now? I didn't know that was wrong.
Can we stop this?
This is the process of gene therapy.
Are you ready for your procedure, Ricky?
Yes.
Are you ready to get that bubble off you that you've been living in for your whole life?
Sure am.
Well, we'll go get your operating room ready, and we'll come back for you in a few minutes.
You stay here.
Sounds good.
All right.
Ricky, we're ready to operate.
Let's go, buddy.
this could be the last time you ever see him.
Good
Right
luck.
this way. You can go wait in the waiting room.
Oh.
Just so nervous. The body could attack the virus and cause problems. The virus might not work. It might attack other parts of the body. Man, if it works though, his life will be so much better. He'll be out of the bubble. My best friend's life will just be so much better.
The operation is over. You can go see him now.
Fine.
Randall, this is the moment of truth. Would you assist me, please?
He's alive! Yes!
Don't go into my operating room!
Sorry. Yeah, buddy, you're alive. You made it. Thanks, buddy.
Finally get to sit in the front. Awesome. Here you go, bud.
That procedure went well.
It did. He's still alive.
This is why we should keep exploring gene therapy.
So people like Ricky can live better lives.
And we can help them out a lot.
Yup. That's our project on gene therapy for Mr. Lauer.